grade 12 learners. Welcome to today's Life Sciences lesson. My name is Samantha Raghunanan. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. You may send messages via our WhatsApp line on the number 079 454 9535. The number again 079 454 9535. You may send messages but not videos or voice notes. Let's get started. So grade 12s, today we are going to do a special session. It's a revision session for the June exam that's coming up. We are going to cover the topics that are going to be covered in the June exam. So let's look at the breakdown of the topics. So firstly, if you look at the weighting on the screen in front of you, you will see that it looks a little different. In your exam guideline, you have topics and you have the amount of marks that they count for in paper 1 and they count for in paper 2. Today's lesson is a little different because it is streamlined to suit the mid-year exam. So the mark allocation will change just a little bit. So let's look at what you need to study and how many marks is each topic in the mid-year exam. So firstly, the topic that we are going to cover is the DNA code of life, which is for 20 marks in the mid-year exam, and then we're going to cover meiosis, which is 16 marks, reproduction in vertebrates, which is 6 marks, excuse me, human reproduction, which is 31 marks, genetics and inheritance, which is 36 marks, and responding to the environment in humans, which is 41 marks. Now, if you look at that weighting of the marks in the examination, you can already start to strategize and navigate how you are going to study for this exam. Obviously, you are, based on past experience and looking at the weighting grid and in the exam guideline, you know you need to focus more on those topics that have a greater amount of marks. So, instead of spending extra time on reproduction on vertebrates, which is 6 marks, yes, you must study it, but I would devote a lot more time on responding to the environment, which is actually 41 marks, and human reproduction, which is 31, and genetics and inheritance 36, and DNA code of life. So you need to look at your list and then decide how you're going to study. So what would we need to study this program? You need the DBE study guides, the SIP material. So when you go for Saturday SIP or when the SIP material is supplied to your school, you should be filing that material and using it for study purposes. Past year papers, prelim papers, teacher notes and activities, exam guidelines, Mind the Gap, which is very, very important. We know Mind the Gap is simplified so that it's easy for us to understand, lots of clear-cut diagrams, lots of labels, and easier for us to take the information in. And then you've got the department's own website, which is www.education.gov.za, and then you've got the Pocket School website, which you can also go to. So let's get right into it. So firstly, we're going to touch on DNA. There's certain key points to remember. Yes, we know that we always go back to our exam guideline. For the purposes of today, the exam guideline has been summarized here, and we see that we have the history of the discovery of the DNA molecule. So you need to know the history, the role that Watson, Crick, Wilkins, and even Rosalind Franklin, what were their roles in the discovery of the DNA. Structural difference between the DNA and the RNA. When we say structural difference, diagrams, okay? The differences between DNA, messenger RNA, and transfer RNA are important. We need to be able to distinguish between them. Differentiate between DNA replication and protein synthesis. This is a popular question, which we will revisit in a short while. Transcription and translation must be learned by heart. When I say it must be learned by heart, you need to know it exactly as it is from the exam guideline. Textbooks have various versions of them, but it's safer to go with the exam guideline because that is what the examiner is going to use when they are setting the marking guideline and when the markers are marking. And then you have DNA profiling. It's important for application questions. Paternity testing involves both parents and child, and the uses and the interpretation of DNA profiling. So under DNA Code of Life, you need to know all of these points, and you need to be able to answer questions based on them. So the first point that we covered was the structure of the DNA and the RNA. So if we look at these two structures, we can already see that there is a difference in the structure. We know that the sugar in DNA is called deoxyribose, and we need to ensure that we know how to spell deoxyribose, because a lot of learners make the mistake. We know that with some of us, English is not our first language. We have to spend extra time on terminology so that we get the spellings correct. So when we talk about deoxyribose, make sure that it is spelled correctly, because when the spelling is incorrect and it gives a different meaning, it is marked wrong. Okay, so there you have the DNA structure. We know that it is double helix. You cannot say double-stranded. You cannot say anything else. The only acceptable description for the structure of DNA is double helix. It is long and it is made up of monomers called nucleotides. And then let's look at the RNA, because we put them side by side so we can compare. Can you see the structure of this is very much different from this one? Here you have a single strand. There you have a double helix. And we know that we've got the sugar, which is a ribose sugar. It's a single strand and it's shorter than the DNA strand. So you need to be able to label the structure. You need to be able to identify and distinguish between DNA and RNA. And we know that it is made up of nucleotide bases, which we are going to come to in a very short while. Let's isolate RNA structure. RNA is a single-stranded molecule. It is made up of a pentose sugar and a phosphate group, which you would find there, and 4 nitrogenous bases. So these nitrogenous bases are specific to RNA. So you have adenine, which is abbreviated A, uracil, which can only be found in RNA, and guanine and cytosine. So I've highlighted uracil because uracil can only be found in RNA. When it comes to DNA, we will find thymine in DNA as a nucleotide base that pairs with adenine. Now, when you are trying to identify a structure, you first need to establish whether it's DNA or RNA, and you can look at the structure of it, whether it is double-stranded or single-stranded, whether it's got uracil or whether it's got thymine. Those are clues that will allow you to identify your diagram. So there's 3 types of RNA. You've got messenger RNA, which is known as mRNA. We've got transfer RNA, and we've got ribosomal RNA. So if you want to write those abbreviations, it is acceptable in the exam to write those abbreviations. Okay, moving on. Let's talk about DNA replication. Now, DNA replication, when learners look at this diagram, they become so confused. They look at the diagram and they become overwhelmed. It's actually very, very simple. And DNA replication is an important process because it must happen between before mitosis and meiosis. It is important for cell division to occur. So let's talk about DNA replication and what exactly happens in DNA replication. But before we go to the notes, let's just visit the diagram and see what we can make out of the diagram. Because sometimes it's easier to learn with diagrams than just from the notes. It's easier to look and see and observe. So if we look on the top here, we can see that we have a DNA molecule. Can you see that? There's the DNA molecule. And we know that the structure already, we said double helix. We must keep remembering this double helix. And as we can see, it unwinds. So it is like this, and then it's unwinding. Can you see that? There's it unwinding. And when it unwinds, it then unzips. So if you look at unwind and unzips, sometimes learners confuse the two. They'll say it unzips and then it unwinds. How do we remember this? Remember, W comes before Z. So try and remember that it's actually in alphabetical, okay? So that's how I remember it. So there you got the DNA molecule, it unwinds and then it unzips. When it unzips, you'll see what is happening to the nucleotide bases. They are separating. Can you see there's a space that's forming there? Because they are unzipping. When they are unzipping, the nucleotide bases are separated. Can you see that? And then you can see there's free nucleotide bases that are floating around here. And we're not going to go into too much detail, but we're going to look at the diagram. And then you can see something else happening here at the bottom. Can you see? They're starting to reform again in their complementary base pairs. And if you look at the bottom of the diagram, you have two identical DNA molecules. I want you to remember that it ends up with two identical DNA molecules, not DNA strands. DNA molecules. Yes, two strands are formed. There's the two strands. But the end result is two DNA molecules. So let's go to the notes and quickly go through the diagram. So there we have it. The double helix unwinds. Number one. Weak hydrogen bonds between the nitrogenous bases break and the DNA, the two DNA strands unzips. So this point is quite significant. Weak hydrogen bonds between the nitrogenous bases. So in order for the nitrogenous bases to be joined together in their complementary base pairs, the hydrogen bonds helps it to keep together. But because these hydrogen bonds are so weak, it enables it to separate, and that is how it is able to unzip. Okay? And then another word for unzip is obviously to separate. And then we have each original DNA strand serves as a template on which the complement is built. Here's the original DNA strand, and there's the second original DNA strands. Okay? And three nucleotides build a DNA strand onto each of the original two strands by attaching their complementary nitrogenous base A to T and C to G. Remember, A is complementary to T and C to G. And because it is DNA, we are talking thymine. Okay? So that is basically what happens. So point number 4, complementary strand forms. You can see there is C, there is G. It's complementing each other. So they are joined there. And if you look at number 5, this results in two identical DNA molecules. So as you can see, that is how DNA replication takes place. Each molecule consists of one original strand and one new strand. Let's move on to the next process, which is protein synthesis. Now, protein synthesis is an important process because proteins are the reason we have hormones, they're the reason we have enzymes, we have antibodies, all of those things. So the production of protein is an important process to be studied. So it takes place in two places. The protein synthesis takes place in the nucleus and in the cytoplasm, namely at the ribosome, at the site of the ribosome. So let us look at the two phases that it takes place in. Now, if you look at the transcription, the first phase of protein synthesis is transcription, and it takes place in the nucleus. So let's look at the diagram. The double helix unwinds. There's your double helix. Can you see? Okay, and it unwinds. There's it unwinding. The weak hydrogen bonds between the two nitrogenous break and two strands unzip or separate. There they are unzipping and they're separating. So it is very, very similar to DNA replication. But that's where the similarity ends. So please do not confuse DNA replication with transcription. It's two completely different processes. You will see why in a short while. Now, if you look at point number 3, one DNA strand acts as a template for the construction of messenger RNA in the nucleus. One DNA strand. Already there's a difference because for DNA replication, we have two DNA strands that acts as a template. Here we just have one. So basically what happens in protein synthesis, the DNA has a formula, it has a code, and this code needs to be taken out into the ribosome, into the cytoplasm, but the DNA doesn't leave the cytoplasm. So therefore, it uses the transport of the messenger RNA to go through the pore and go out and get the message out, so that that code can be transferred and reproduced to produce more amino acids. So there we have it. You have the free nucleotides. From the nucleoplasm, they form messenger RNA. Here's your messenger RNA. So messenger RNA basically is exactly as the name says, it gives a message. So there we go. So there we have the codons, the three triplet bases. They form the nucleotide bases form codons on the messenger RNA. It leaves, and the coded, and then it goes into the ribosomes. So at the ribosome site, it leaves the nucleus through the nucleopore to the ribose. The transfer RNA, this diagram here is the transfer RNA. You need to be able to identify what a messenger RNA looks like, what a transfer RNA looks like. Why do you need to be able to identify that? Is because in the exam, they will give you a diagram. They're not going to tell you, this is the messenger RNA, this is the transfer RNA. Maybe if you're lucky, they will. But most of the time, a diagram is given and you have to identify, this is where transcription takes place. This is where translation takes place. How do I know this? Because this one is taking place in the nucleus. That one is taking place in the ribosome. So that is how your logic must work, because when you know the content and you know the diagrams and you know the terminology, you are able to distinguish them in the exam. So there's a transfer RNA. It's a hairpin-like structure. It's got the amino acids on the top, which is the building block of the protein, and it's got the anticodon. And we know, because this is a revision lesson, am I right, grade 12s, that at the bottom of the transfer RNA, we have got a triplet anticodon nucleotide base, which is complementary to the messenger RNA. And the transfer RNA pick up the amino acids and they complement the messenger RNA in the ribosome. The amino acids bond by a peptide bond to form a protein. There's the amino acids that are bonding by a peptide bond. So that's basically a quick revision of how protein synthesis takes place. So key points to remember is you may be able to identify the site at which transcription is taking place and translation. You also need to distinguish between DNA replication and protein synthesis, knowing that in DNA replication, one strand is used as two strands are used as a template, and in protein synthesis, one strand. And in protein synthesis, we have 3 RNA nucleotides, whereas in DNA, we have DNA nucleotides. Okay? So those are key points for you to remember. Now let's talk about DNA furthermore. So DNA profiling is something that you've come across. So it's a technique used to identify a sequence of bases. So basically what DNA profiling, it's fragments of DNA in the form of light and dark bands of different thicknesses, of different lengths, and it is used to identify. We know that DNA is specific to every individual. So this DNA profiling is used in many different areas of forensics or in genetics, where we are trying to trace a lost relative, where we need to match the DNA to see if people are related. If DNA is found at a crime scene, we will take the sample and we will match it to the bands of the suspect's DNA, and that is basically how DNA profiling works. So this entails the nucleotides being separated from each other in order that they are found in a strand of DNA. Nucleotides appear as dark bands, and DNA profiling is different from fingerprinting. So we should not even be talking about fingerprinting, because fingerprinting, there is nothing in the exam guideline that says anything about fingerprinting. Everyone has an individual fingerprint, yes, but it's not for the purposes of the matric exam. So what you need to be able to do is you need to be able to identify, like you see in this diagram, at the crime scene, there is the DNA sample, and there is the suspect's samples. So we need to learn how to read these samples. So when you go into the examination room, what you need to obviously have is a ruler. you are going to compare the similarities between the band of the original or the control model and the bands of the suspects, as in this case. So for example, you will take a ruler and you will draw lines through common areas where they match. Now you can't draw it like this. You will need to use a ruler, but you can already see that some of the bands they match, others do not match, and the most number of bands they have an equal or closest to the number of bands of the original would be the match for the DNA. So let's just go back a little bit. You might get an exam like what happened in the May-June last year, where they give you a DNA profile and then they give you the girl, her mother, and 4 male samples of their DNA profiles, and they ask you which one is the girl's biological father. That's when you're going to have to start to match the bands. And if we match the bands, we will see that the girl matches. If we draw the lines that are going from the girl, it is quite clear cut when we see that the answer is obviously C, which is number 3, because can you see the bands are already matching here? 1, 2, 3. So that is how you determine DNA profiles. Okay, let's move on to meiosis. So meiosis is a reduction division. It is a very important process, and we need to know in meiosis, we need to know the different phases of meiosis. So they don't usually give you a paragraph question. Most of the time it's a diagram, and a key to understanding meiosis is learning the diagrams, being able to identify a diagram and which phase it represents. So from the exam guideline, you need to know that there is a production of gametes. We know that meiosis is usually, the purpose of meiosis is for the production of gametes. But before meiosis, the cell prepares, excuse me, before meiosis, it begins DNA and replication. DNA replication must take place. This results in each chromosome containing 2 chromatids joined by a centromere. Okay, we've already touched on this, but we need to know the whole preparation of DNA replication, which we've already touched on. And then meiosis is divided into two parts. There is meiosis 1, which is the first division, and there's meiosis 2. And in meiosis 1, we have 1 diploid, which gives rise to 2 haploid cells. In meiosis 2, we have 2 haploid cells, which give rise to 4 haploid cells or gametes. So that is a distinguishing feature between meiosis 1 and meiosis 2. One of the main importances of meiosis is the biological importance, because crossing over takes place. Now we know cells divide, but you cannot have clones walking around everywhere. Therefore, we need genetic variation. That is why we look different from our parents. We are not clones of our parents, because we must ensure that there is a variety of species in the coming offspring. And we also need to make sure that the ploidy number remains constant, so that we don't have any extra fingers or toes, and that the chromosome number is remaining constant. So when it comes to the terminology with regard to meiosis, we have different terminology which learners tend to confuse. We're not going to go through each of them since this is a revision session, but we need to know, for example, homologous chromosomes. They are identical chromosomes. The genes are found at the exact same site. These are identical chromosomes. Bivalent is when they come to lie next to each other and crossing over occurs. And then we need to know the difference between centromere, centrosome. We need to know the difference between karyokinesis and cytokinesis, because it sounds so similar. So that is where you need to learn your definitions. So what I'm saying now in the revision for the mid-year exam, you need to learn your definitions and your terminology, so that you are able to distinguish terms which will come out in section A. Let's talk about the different phases of meiosis. Now we talk about interphase. Interphase is the preparation phase. This is where DNA replication occurs, And we have two chromatids which are joined by a centromere. You should know this by now. DNA replication helps to double the genetic material so that it can be shared equally. This is the main reason. So it can be shared equally during cell division. So two terms that are very, very important that you can see on your screen is a unreplicated chromosome and a replicated chromosome. It's so simple. You just need to look at the diagram to know that the unreplicated chromosome would be the chromosome that is present before DNA replication, because there's just one chromatid there. You have a single chromosome. And then you have the replicated chromosome, which is a double chromosome, 1, 2, where two chromatids occur after DNA replication. So that is the difference between the two. So let's talk about the phases of meiosis. So when we were doing the section on meiosis, whether it was in grade 10 or earlier this year, we knew that we used the IPMAT system where we learned about the different phases and learned to distinguish. So we're going to do a summary of each phase and to see how we know the difference in the exam. So if you have a diagram like this, it can be very, very helpful. So let's talk about prophase 1. What is distinguishing about prophase 1? Prophase 1, we know that is where crossing over takes place. The homologous chromosomes come to lie close to each other and crossing over takes place. Okay, so that is the distinguishing feature of prophase 1. It is different from prophase 2. Can you see prophase 2 here? Prophase 2, how would we know that a phase is in prophase 1 or prophase 2? Prophase 2 has a patchy appearance. And by the time prophase 2 takes place, crossing over has already taken place. Okay, so that is how you're going to distinguish it. And then we look at metaphase. Meta, remember meta meaning middle, isn't it? Middle. But we don't use the word middle, we use the word equator. So in metaphase 1, if we recap, we will remember that the pairs of chromosomes align themselves at the equator randomly. Remember, we said that this is very randomly, and this was the reason that we have genetic variation, because of the random arrangement of these pairs of chromosomes at the equator. But let's look at metaphase 2. Metaphase 2 down here, there's metaphase 1 and there's metaphase 2. Metaphase 2 is very, very easily distinguishable. We got a single a group of singly arranged chromosomes which arrange themselves along the equator. So you'll be able to distinguish metaphase 1 from metaphase 2 because of the single arrangement in metaphase 2. Now let's talk about anaphase. Anaphase, ana meaning away, they're moving away to the poles, okay? But if you look at anaphase 1, we know that in anaphase 1, they are moving away to the poles, but these are whole chromosomes that are moving towards the poles. Can you see there? Whole chromosomes, they are moving towards the poles. Anaphase 2 is different. Let's look at anaphase 2. Here we have chromatids. The chromatids are single. And what happens here? In anaphase 2, it's different from anaphase 1 because remember the centromere splits here. And when the centromere splits, the spindle fibers shorten. There's the spindle fibers. They shorten and they cause single chromatids to move towards the poles. So that is the difference between anaphase 1 and anaphase 2. So we can see that we can use our knowledge of meiosis and we can summarize the main distinguishing features of each phase so that we are able to answer any question that they pose to us in the exam. So let's talk about telophase. Telo, remember we said it's 2. Okay, so here in telophase 1 we see the invagination occurs and it results in 2 cells, and in telophase 2 we know that we have 4 haploid genetically different cells. So these are all the different phases of meiosis that we are able to identify. You can print a diagram like this and you can put it up and make notes, and it's like a one-pager for you to revise for the exams. So let's look at the May-June paper, we're going to quickly go through examples of these so that we know what sort of questions they can ask in the exam. They ask you here, identify the phase shown. Now, before you even look at the questions, you must look at the diagram. If you look at the diagram, you will see that something is happening here by the equator. I already know it's something to do with metaphase. But I know it's either metaphase 1 or metaphase 2. But because it's a single arrangement of chromosomes, I already know that the answer would be metaphase 2. One observable reason, I have just highlighted that it's individual chromosomes that line up at the equator. Identify the structure A. A is the cell membrane. This is an easy question, okay? And B would be obviously the spindle fiber. Describe the role of the part B in the movement of the chromosomes during meiosis. So it contracts. We know that when the spindle fiber contracts, it shortens and it pulls the chromosomes or the daughter chromosomes. Remember, we call them daughter chromosomes as they are moving towards the poles. So that is the role of B, and B being the spindle fibers. So if we continue, here you have a diagram of, we can already see, when we look at a diagram, the first thing we must try and distinguish is what phase is this. The minute I see crossing over, I already know it's prophase 1. And you should too. So if we identify the structures, we know that that's a spindle fiber, we know that that is a centromere, and we also know that that is a chromatid, okay? Which phase? We've already discussed that. We know it's prophase 1. Don't just write prophase, you're going to get it wrong. Because remember, there's prophase 1, prophase 2. Prophase 2 is a patchy appearance. So now you need to describe the process of crossing over, and we know that the chromosomes come together to form homologous pairs. They overlap. There you see them overlapping, and the point at which they overlap is called the chiasma, and genetic material is exchanged. This is all revision, so we're not going to be able to stop at every stop, okay? We have 3 reasons why meiosis is biologically important. This is important because we need to have gametes, and we need to maintain the chromosome number, and we need to have genetic variation. This is work that you have done in grade 10. So lucky for you, it is just a revision. And then we have another question on meiosis. Basically, you just need to draw a diagram here of the part that is undergoing crossing over. If we move on, we talk about non-disjunction, and we know non-disjunction is when there is when the spindle fibers fail to separate during meiosis. So if there is non-disjunction in chromosome pair number 21 in humans, it leads to the formation of an abnormal gamete, and we know this as Down syndrome. If we move on, there's the results of non-disjunction, where you have normal separation and each chromosome goes into a gamete, but here where they fail to separate, both goes into one gamete, and this gamete is left without a chromosome. So that is basically the results of non-disjunction. So let us move on to reproduction in vertebrates. Because this is only 6 marks now in the June exam, you basically need to know the difference between external and internal fertilization, ovipary, vivipary, and ovovivipary, the amniotic egg, precocial and altricial development, and parental care. So if we talk about external fertilization, what you need to know is external fertilization needs a lot of water. It needs water for fertilization to take place. Internal fertilization is in a controlled environment inside the organism's body. So we're going to talk about the advantages and the disadvantages of each. So if we look at external fertilization, you don't need additional energy because there's no parental care involved. The eggs are laid and then the sperms are deposited over the eggs, so there's no parental care. No need for the male to have a special organ to insert the sperm inside the female's body. Remember, even with the fish, the female lays the eggs and the male comes and deposits the sperm over. So he doesn't need to insert it inside the female's body. Chances of fertilization are enhanced by courtship displayed by fish. You know the fish do the wiggling, so there's courtship behavior, so this helps for external fertilization to take place. Okay, the disadvantages of external fertilization. Remember, if the organism is just laying eggs there, anything can happen. Weather conditions, floods, currents, all of those things, and predators can even come, as you see here, and they can destroy these eggs. So the chances are very slim for fertilization. There's a lot of external factors which can interfere with the fertilization process. Okay, if we go to reproduction, like I mentioned earlier, external fertilization takes place outside the body. So reproduction must take place in water. And if reproduction doesn't take place in water, then the eggs, or the ovum is going to dry out. Remember, we don't ever say egg cell, we say ovum. Okay? And then let's talk about internal fertilization. Internal is obviously inside the body. It ensures that the sperm cell comes into contact with the ovum and the developing embryo is protected from predators. Because the sperm is being deposited into the female, that female is protecting that fertilized ovum. Okay? The disadvantages is that you can only produce fewer eggs in internal fertilization, and you must have a copulatory organ. As you can see there, with these chickens, it has a copulatory organ where the sperm is deposited into the female. So if we move on to ovipary, now we know ovipary is when eggs are when the eggs are released and they are hatched and the organism is produced. Ovovivipary, we know that ovovivipary, as a revision purposes, is where the egg develops inside the body and it hatches inside the body and the young are born. So what are the advantages of ovipary? Eggs and sperm cells can be produced in large numbers. And then you have parental care. Okay, there you can see where the eggs are laying, you've got the little birds, and obviously the mama is going to come and bring all the worms there. So there you have parental care, that's the advantage. But if we look at ovovivipary, let's look at the snake, for example. Now remember, the eggs are hatched inside the female's body and there's the young born. They are protected from the cold and from predators. Obviously, they are in a controlled environment. The young are well developed and they are born, so the chances of survival increase. Because they have time to develop inside the female's body, they are strong enough when they are born. So embryonic development, vivi, alive, isn't it? Please don't confuse these terms. These are easy marks for you to get. So vivipary is where the young are born alive. And the advantages, it occurs inside the mother's body. The young are well developed. Look at this little elephant there, baby elephant. The parental care increases and the survival chances of the young are improved. The disadvantages of vivipary, you have a fewer number of offspring. And remember, it involves parental care. So parental care takes energy, okay, because you need to feed the offspring, you need to make sure that they are warm and all of those. This is the example of an amniotic egg, and we know that the amniotic egg, what we need to remember about the amniotic egg, it's got the yolk which provides nutrients, it's got the amniotic fluid which protects the embryo. Remember, the amniotic fluid is like a shock absorber which prevents against mechanical injury. And you have the chorion, which supplies oxygen, food, and water. There's the chorion found there. And then precocial and altricial development. So we're going to look at this as a whole. Precocial, we always say precocial meaning prepared. The organism, the offspring is prepared. That means they hatch with their eyes open, they have soft feathers to keep them warm, they're able to move soon. So we say that it's precocial because the young are highly developed. That means they can take care of themselves as soon as they are born. So precocial, they are prepared. Altricial is the opposite of that. If you look at the baby, as soon as the baby is born, the mom needs to wrap the baby into a warm blanket, provide temperature control, he needs to feed the baby, all of those things. So they need all care from their parents. And they do not have feathers, they need to be fed, they need to be kept warm. So that is how you will remember the difference between precocial and altricial development. Now, in our next part of our work, we're going to talk about human reproduction. And I'm going to cover the female and the male reproductive system. So if you look at the female, you need to know the structure of the male reproductive system. So there's many parts to this male reproductive system. If you look at the accessory glands alone, we've got the prostate gland, the cowpus gland, and the seminal vesicle. You need to know what is required by your exam guideline. So therefore, you need to refer to the exam guideline, and you need to be able to label the front view and the side view. Okay? And connected to the male reproductive system, a popular exam question is the sperm cell. A reminder again, the sperm cell, the shape of the sperm cell is torpedo. It's not oval, it's not round, it's torpedo shape. It's got the mitochondria, muscles, energy, so it enables it to have energy for locomotion. It has got the long tail, and the long tail, we know, enables it to swim up the fallopian tube. The acrosome on its own has digestive enzymes which help to dissolve the ovum's membrane. And then we talk about the female reproductive system. The female reproductive system, the same. There are many labels. You need to go to your exam guideline, look at your Mind the Gap, and learn to label all of these diagrams, because you can be asked to label all of these labels. You know the structure of the female, you know the fallopian tubes, and you know that in the fallopian tubes we have hair-like structures which enable the ova to move, and we know that we've got the uterus where implantation occurs, the cervix which opens out, the vagina which is the passage which opens out into the vulva. Key to the female reproductive system, boys and girls, we need to know that the ovum is a structure which you need to know how to label. So with regard to the ovum, you have the cytoplasm, and we know that the cytoplasm has nutrients. So in the cytoplasm, this nutrients for the future fertilized zygote, this cytoplasm is going to provide nutrients. We got the nucleus, which is going to provide genetic material. You need to be able to draw this diagram, you need to be able to label the diagram, and answer any questions which is based on this diagram. With regard to the type of question you could get a structure like this, where you would obviously need to label. You can see there, B is the seminal vesicle, and then G is the urethra. And then they would also ask you, where is the sperm cap mature? In the epididymis, which is D. And what controls the temperature where the sperm is produced? And that would be obviously the scrotum. So we're going to pause there for a while and take a short break, and we're going to be joined by my colleague Vedika Maraj, who is going to take you through to the next part of the revision program. Stay tuned.
Welcome back, grade 12. I just want to remind you before we carry on that we have a WhatsApp number 079-454-9535. Also that this is a revision lesson, so we will not be going through the details of the topics, but just an overview. But if there are certain topics that you know that you're having problems with, or you are struggling with, then you can revisit our lessons that we did earlier on in the year, because as you know, we did it according to the ATP. Every week we had a lesson on a particular topic, so you can go and revisit those where we go into more detail. Oh, and by the way, my name is Vedika Maraj, and I'm going to take you through the rest of the content that's needed for the June exam. So here we have the menstrual cycle. The main points that I need you to understand about the menstrual cycle is that you need to know that it's made up of 2 cycles, which are the ovarian cycle and the uterine cycle, and you need to know the changes and the events that occur in these different areas. So the ovarian cycle has got to do with ovaries and the developing follicles, and within the follicle we have an ovum. And the uterine cycle has got to do with the uterine lining, or the endometrium. Basically, it's got to do with the uterus, and the uterus has an endometrium, okay? And this endometrium changes over the 28-day cycle, and as well as the follicle develops over the 28-day cycle. Now, that's not the only thing you need to know. The next thing that you need to know are the hormones, okay? So we have a little acronym here to help you remember the hormones. We have F, which is FSH, O, estrogen, L, LH, and P, progesterone. And you can look in the diagram. It is showing us that LH and FSH are pituitary hormones. So they are secreted by the pituitary gland, and these secretions are going to affect what is happening to the ovary, okay? What is happening in the ovary. So it comes from the brain, from the pituitary gland, but it affects the ovary. That is the target organ. Whereas the ovarian hormones are the ones that are secreted by the ovary. Those are estrogen and progesterone, and those are going to stimulate other hormones, as well as they're going to affect the changes in the endometrium. Okay, so this diagram is a very nice one to explain everything in one. So when you are learning, I'm sure most of your teachers have done this with you. You also can refer back to our previous lesson where we did this. Okay, we're going to take a 2-minute break before we go to the flow chart.
You have been given power to decide whether you want good results in your life, in your schoolwork, or you want bad results. As Tony Robbins said, it is in the moment of your decision that your destiny is shaped.
chemical glitch. Let's get back into it. This is a flow chart summarizing the interaction of the different hormones. Please study the slide very carefully. It gives you a little bit more information on what I was discussing in the previous slide, but I really want to do the activity that I have prepared for this section, which is going to help you sum up everything nicely. So this is an extract question. Please remember that it's very important as you prepare for the exam that you do lots and lots of past paper questions. It's not only about knowing the content, as you are going to be faced with these questions. And life sciences loves extract questions. Usually an extract is about something that you have studied, but it gives you added information. This one is about plastic linked to female infertility. Several studies indicate that bisphenol A, BPA, a chemical used in the production of many household plastic products, may be linked to female infertility, which is the inability to get pregnant naturally and to deliver a healthy baby. BPA can be ingested or absorbed through the skin when using plastic products. BPA seems to interfere with the normal secretion of FSH by the pituitary gland and is linked to abnormal menstrual cycles and reduced implantation rates. These studies also show a link between high BPA levels and a decrease in the development and maturation of ovarian follicles. Okay, so you can already see from this information that this is affecting the menstrual cycle. So when you read, you must read with understanding. And you must be able to figure out what is it about. So you need to read it about twice, okay? And also you should read it once, then read the questions, and then read it again, so that your mind can already start processing the answers to the questions. Okay, so the first one is stating the function of FSH. Now this did not, it does not tell you, okay? It just tells you here that it interferes with the normal secretion of FSH, but it doesn't talk about how it's going to affect it or what is the function. So you should know from your studies that it stimulates the development of ovarian follicles and it initiates puberty. If I wake up in the middle of the night, you need to tell me what are the functions of the 4 hormones: FSH, LH, estrogen, and progesterone. Name one other hormone in females that is secreted by the pituitary gland during the menstrual cycle. So you see they're referring you to the pituitary gland. So then you know if you're talking about FSH in the previous question, now we are talking about LH. Explain how an undersecretion of the hormone in question 2 may lead to infertility. Now, the first thing I want to alert you to is that explain means that you must have an answer as in a cause and effect manner. Okay, so you need to give us a cause and you need to give us an effect. It is 2 marks. 1 mark for the cause, 1 mark for the effect. You need to tell us how an undersecretion of the hormone leads to infertility. So you need to know what is infertility, and you need to talk about if there is low LH, what is going to happen? How does it lead to infertility? Okay, so the first thing you should tell us is that what does LH do? Okay, it stimulates ovulation, right? We know that. Therefore, ovulation will not take place. Why is ovulation not taking place? Because remember, we have an undersecretion. Okay, so LH stimulates ovulation, but there will be no ovulation taking place because LH levels are low. You can also include that to finish your sentence. And there will be no ovum to fertilize. Okay, it's important that you also mention that point. It's only for 2 marks. If you just say LH stimulates ovulation, therefore ovulation cannot take place, you will get your 2 marks. But some learners will also go further to say that there will also be no ovum. And if there's no ovum, fertilization cannot take place and it leads to infertility. Explain why. Another explain question. Oh, I just wanted to show you where is the, okay, we'll do it when we do this one. Why a decrease in the maturation of ovarian follicles may lead to reduced implantation rates. So now we've got decreased maturation of the follicles. So the follicles are not going to mature. And what is a mature follicle? It is a Graafian follicle. And where is the ovum? Inside the follicle. So if the Graafian follicle is not going to develop, what's going to happen? Let's just think about it. The Graafian follicle is not going to develop. And what does the Graafian follicle secrete? The Graafian follicle secretes estrogen. What does estrogen do? It builds the endometrium, right? So if there's going to be no Graafian follicle, there's no estrogen, and there's going to be no endometrium for the embryo to implant, or the blastocyst, to implant, right? The other thing is that we might not even have an ovum because we are not having this proper follicle maturation. Okay, so let's look at the answer. We have a Graafian follicle is not formed. I mentioned that since the Graafian follicle secretes estrogen, estrogen levels will be reduced, the endometrium will not develop, and no implantation can take place. Okay, so you need to mention all of that. So what is the cause? The cause is that the Graafian follicle is not formed, okay? And because the follicle and it secretes estrogen. So you mentioned all your points. And what is the effect of this? The effect of this is that the endometrium will not develop and the implantation cannot take place. So these ones are the effect, and the other 3 points are the cause, right? Then the other point, so you can either talk about how there's going to be no endometrium, or you can say there's no ovum produced, or Graafian follicle is not formed, so ovulation does not occur. So there can be no fertilization. And if there's no fertilization, there's no zygote that's formed. If there's no zygote, it's not going to develop into anything. And then therefore there will be no implantation taking place. Okay. So this is a twofold thing. It's answered in a cause and effect manner. Please make sure you just keep your ducks in a row. When they ask you this question, just go to the question. What are they asking me? Write down all your points and give us a reason why that is not happening. Right. Then we move on to, that's the menstrual cycle. Now, there's something that we need to understand about the menstrual cycle that I didn't mention, but you know this from your class, is that the whole point of the menstrual cycle is to prepare the body for possible pregnancy, okay, and for gestation. So what actually now happens once the ovum has been ovulated, the ovulation occurs and the ovum is now sitting in the fallopian tube, it's going to patiently wait for the sperm to swim up and to fertilize the ovum. So you need to know what happens in fertilization, okay? You need to know what happens to the development of the zygote and implantation, and then you need to know about gestation. Okay, so this part, the most important thing I want you to understand here is that we have fertilization taking place, where we have the haploid nucleus of the sperm cell, which is going to fuse with the haploid nucleus of the ovum to form a diploid zygote. Very important definition that you need to know. Okay. And then this zygote is now going to develop, and it's going to develop into other structures by a process called mitosis. Okay, so mitosis is going to happen. The zygote is then going to divide into a 2-cell structure, a 4-cell structure, an 8-cell structure, and then a solid mass of cells, which is called a modula. It then further divides to form a blastocyst, which is a hollow ball of cells, and it's filled with fluid. Okay, then that blastocyst is going to move down the fallopian tube, move down the uterus, and it's going to implant. Okay, it's going to implant. And how does it implant? It implants because now this blastocyst is going to be having different layers, which are called extraembryonic membranes, right? So you need to know them. What are they? They are the amnion. They are the chorion. The chorion is the outermost. Let's start with that one. It has the chorion. That's the outermost layer. Then we have the amnion. We've got an amniotic sac, which has amniotic fluid. Okay. And then we need to talk about how this implantation takes place. You need to know that the chorion develops chorionic villi, and the chorionic villi embed into the endometrium, and there is no mixing of the blood. Okay, between the maternal and the fetal blood, there is no mixing, because these chorionic villi actually embed into the endometrium, so into the sinuses. The endometrium is now surrounding these chorionic villi, and we just have our diffusion of our gases and our nutrients. Okay. Another important part that I want you to understand about this is that chorionic villi together with the endometrium forms the placenta. The placenta is a very important structure. I'm going to remind you about what is the function of the placenta, but please don't forget about the placenta, and you need to know other functions of the placenta. Then there has to be a way that the fetus is going to get nutrients, right? And you know that the nutrients come from the mother, so you need to remember this very important structure, which is called the umbilical cord, which has 2 umbilical arteries and 1 umbilical vein, and this is what's going to allow the fetus to have oxygen and nutrients, and it's also going to dispel the carbon dioxide and the waste products to the mother, okay, so that the mother can expel it out of her body. Right, please study this summary. They love to ask, it says in your examination guideline, you need to be able to know the structure of the gestation with the fetus inside the uterus. So if you get this diagram, you need to know how to label it. Okay, you need to know the functions of the amniotic fluid, the amnion, the chorion, the placenta, the chorionic villi, and the umbilical cord. Okay, please look at the slides that we did earlier that give you more information of these, but this is a very nice summary for you to study for those of you that enjoy studying from summaries. Right, then we move on to genetics and inheritance. These are just highlighted straight from your examination guideline, so I'm not going to spend a lot of time on it. I'm just going to mention the headings for you. You need to know the concepts of inheritance, okay? You need to know Mendel's laws. You need to know the monohybrid crosses, okay? And what are the monohybrid crosses? Which ones do you need to know? You need to know complete dominance. You need to know codominance, incomplete dominance, sex-linked inheritance, multiple alleles, which is blood groups, okay? And then the next topic is dihybrid crosses. So that is when we have two characteristics. Your monohybrid cross, we are only looking at one trait or one characteristic. Dihybrid crosses, we are looking at two. Then we are moving on to pedigree diagrams, which is your inheritance of your genetic, your family tree and your lineage of how your disorders or how your genetics are basically passed down through your family. And then genetic engineering, which I'm not going to be doing today, but please don't forget about it. I'm also not going to be doing paternity testing. Please do not forget about it. Okay, so I'm just going to go through the other ones very quickly. But like I said, it's just a division lesson. Please do not forget about these things. Study in advance so that you can actually tick off every bullet in your examination guideline, because that essentially is your scope of your paper. Right, then, before we go any further, genetics is two very important things about genetics: terminology and practice. Okay, it's like math. So you need to know your terms, you need to know your rules. You need to know your rules, you need to know your laws, because those are going to help you explain or help you go through your actual questions that they give you. Okay, because you're going to have to draw monohybrid crosses. They're going to ask you questions on dihybrid crosses, and really all of it is practice. There's very little studying actually in genetics. It's all about practice. So you need to know these terms. They are also commonly confused. Chromatin versus chromosome. So chromatin is when they are still entangled. Okay, chromosomes are when they are now the DNA replication has come, they are more condensed, and now they are forming two chromatids with a centromere. You need to know the difference between the gene and the allele. Okay, alleles are different forms of the gene. The gene is the sequence that codes for the particular trait, and every gene has two alleles. Okay, and the allele are basically contrasting characteristics that are going to be making that speak to the gene. You need to know the difference between a dominant and a recessive allele, phenotypes and genotypes, homozygous versus heterozygous, and monohybrid and dihybrid. Okay, so please make sure you know these and don't get them confused. You need to know the three Mendel's laws of inheritance. Please look in your exam guideline on page 11, page 12, and on page 12. You need to know them word for word. They are on the slide. But if you don't have the slide with you later on, make a note for when you're studying that you need to understand these three. It's the law of dominance, Mendel's principle of segregation, and Mendel's principle of independent assortment. Make sure that you know them. These are part of the rules I'm talking about, because they can ask you to state these word for word. So what can they do? They can ask you to state these. They can also ask you as an application question where you need to apply your knowledge and use these rules in order to apply. The format of the genetic cross, remember, you will get a monohybrid cross to draw. You will definitely get a monohybrid cross to draw. Whether it is going to be a complete dominance one, incomplete, codominance, sex-linked inheritance, or blood groups, but you are going to get one. So make sure that before you go into your exam, you practice all of them. Okay, and you practice how to identify which one you are going to be doing, right? So this is the recipe. This is your template or your recipe of the layout of your genetic diagram. Please make sure you know it, okay? We have the P1 phenotype, and then we all know that by now we should know that the visible trait is the phenotype, example, tallness and shortness. The genotype is the genetic makeup, so these are the alleles that are basically coding for that gene. And then we have meiosis taking place, and now we can apply the law of segregation, right? That's the law that I mentioned earlier, where the gametes are now, the alleles are going to now separate during meiosis, and we are going to form gametes, okay? And each of those gametes, and it's important, we spoke about meiosis earlier, Samantha did it, to say that it's going to assist with genetic variation. So we're going to have these gametes that are all going to be genetically different. And what happens to gametes? They fertilize. So what's going to happen here, because one thing I didn't mention is that here we have a father and we have a mother. That is what we are crossing, the phenotype of the father and mother, then the genotype of the father and mother, and the genotype is informed by the phenotype, okay? Or your phenotype, sorry, is informed by your genotype. Your phenotype you will get in the question. Okay? And then we've got the gametes, as we said, and then fertilization. So we're going to have the gametes of the father fertilizing with the gametes of the mother. So the whole point of this is that we're going to work out the probability. Okay? So what is the probability going to be of the F1 generation, which are the offspring, their genotype, because that's what you're going to have first, and then the phenotype. Please remember that the genotype here comes first, and then the phenotype. Okay? So we spoke about all of this, and then this is the other one. If you don't do the cross, you can do the Punnett square. This is a Punnett square, okay? And then when you do a Punnett square, I just want to indicate to you that if you do a Punnett square like this, for example, you put the gametes of the one parent there, the gametes of the other parent there. Please make sure that you have that word gametes in over there, and then you are going to do the cross like such. Okay. So you're just going to cross, you cross this one and this one, then this one and that one, and then you're going to do this one and this one and this one and this one. Okay, and that's how you get the Punnett square. Whichever one you've done, don't try new things now. Whichever one your teacher did with you, that's the one that you're going to do. Remember that by writing P1 and F1 and meiosis and fertilization in the correct sequence, you can get 2 marks. I just want to give you an idea of how you can remember this. Let's start with P. So please, guys, make good friends, via Gauteng province. Okay, so that can give a little acronym for you to help you remember the steps and the different steps that you need to write. If you just write that plus the phenotypes of the mother and the father, you can get 2 marks. Right, now what are the steps to follow when answering a genetic cross? I think I actually went through all of them. So we are going to just go through them quickly. Identify the phenotypes of the parent. Choose the letters to represent the alleles for the gene responsible for the characteristic. Identify the genotypes of the parents. Show how the alleles are separated through meiosis into separate gametes. Remember, law of segregation. And then you need to show all the possible genotypes of the F1 generation when fertilization occurs. And interpret the phenotypes of all the possible genotypes from the cross. Okay, so we're going to do a cross, so I'm not going to focus too much on that slide, because when we do the cross, I'll explain to you what we're going to do. These are the 3 laws of dominance you need to know: complete, incomplete, and co-dominance. Right, when we are looking at complete dominance, it's only one parent's phenotype is evident in the offspring. Okay, so that means that there is one allele that is completely dominant, and that is the one that is shown in the offspring. Unless it's in a homozygous state, in a recessive homozygous state, then it will be shown in the offspring. Okay, so we have one allele that's completely dominant. This means that if I have one dominant allele as the offspring, one dominant allele, excuse me, and one recessive allele, then the dominant allele will be in the offspring. But if we have 2 recessive alleles, we're going to have the that characteristic in the offspring. In incomplete dominance, neither of the alleles are dominant. So neither of the parents' phenotypes are dominant. Okay, and they are not evident in the offspring. Okay, so this is when we have a red flower and a white flower, but the allele for red color and the allele for white color are not dominant. Therefore, we get an intermediate phenotype, which means we got a mixture of the 2 colors. This can apply not only to plants, it can also apply to animals, it can also apply to humans, it can apply to anything, so please remember that. This diagram is showing you pictures, but it can apply to anything. Co-dominance is when we have both parents that phenotypes are evident in the offspring, because here we have 2 alleles that are dominant. Okay, they are equally dominant, which means that if we have a white cow and we've got a red cow, then we are going to have a roan cow. Can you see the color of the roan cow? It's a white cow with like a red color spots. So you can see in the offspring, you can see white and you can see red. So the fact is that we can see 2 colors, which means they are equally dominant and they are both expressed equally in the phenotype. It is not a blended one. Then we move on to sex determination. Okay, so now when it comes to sex determination, we are now looking with the monohybrid casters. We looked at one trait, okay, where we are looking at the color of the eyes or color of or any trait in the human body, whether it comes to any gene or anything, any gene. Remember, genes make up our whole body, so it can refer to anything. But when we are looking at sex determination, I'm introducing you to sex-linked disorders, okay? So let's first talk about sex determination. What's determined a male to be a male and a female to be a female, okay? So you need to know what this karyotype, okay? It's a distribution of the chromosomes, right, in their pairs, the positioning of the chromosomes in their homologous pairs. And you need to know that we have 22 autosomes, 22 pairs of autosomes, which means that we have 44 autosomes. And then now pair number 23 are your gonosomes, and that is either an X and a Y or an X and an X. Okay, these are also known as sex chromosomes. And if you have an X and a Y, then it means that you're a male, and if you have an X and an X, it means that you're a female. What's important about this is that I need you to know what a karyotype is. I need you to know what autosomes are and what gonosomes are. I need you to know how to determine a male and a female. I also need you to know that there's a 50% chance of having a boy and a 50% chance of having a girl. Now, when we're doing monohybrid crosses, usually if we're doing monohybrid crosses of the ones that I was doing now, they are all on the homologous chromosomes, these autosomes. Most of all the genes that we spoke about now are on these autosomes. Okay, but now when we're looking at sex determination, we are looking at the gonosomes. So a female is X and X, and a male is X and Y. And then because of the gametes being formed and the law of segregation and then fertilization occurring, you will see that when you do the cross, you can quickly do a cross for yourself as well, and you'll see that you get a probability of 2 males and 2 females, which means that you have 50% chance of having a boy and 50% chance of having a girl. This is a probability. It does not mean that if I had 2 boys, my next 2 children are going to be 2 girls. It's just about a probability that there's a 50% chance that you're either going to get the X allele or you're going to get the Y allele, because we only have those 2 alleles. Right. Then we go to sex-linked disorders. So like I said, the other genes that we were doing in the monohybrid crosses, that is when the trait is on the autosome. Okay, the gene is found on the autosome. However, in sex-linked disorders, there are still monohybrid crosses, but now the disorder or the gene is found on the X chromosome, never the Y chromosome. It's on the X chromosome of the gonosome, okay, not the Y chromosome. The Y chromosome is very small, so we are not looking at it in our syllabus about what genes are carried there. You just need to know that it's carried on the X-linked allele. Okay, the important thing I need you to know from here, there's 3 take-homes about the sex-linked disorders, or 4 rather. The fact that the gene is located on the X chromosome. The fact that fathers can pass the X-linked allele to their daughters but not their sons, because remember, each parent has to give 1 allele to their offspring. Okay, so if a female can only give X's because the female only has X's, so the mother can pass the sex-linked allele to the father, to the daughter or the son, because she only has X's, right? But the father can only pass his X chromosome to the female because the daughter needs 2 X chromosomes. So the father can only pass that X chromosome to the daughter, and he has to pass a Y chromosome to his son. Then you need to know the 2 sex-linked disorders that's part of the examination guideline. It's hemophilia and color blindness. Hemophilia is when you have a problem with the clotting, with blood clotting, okay, and then color blindness is when you've got red, green, confusion, color blindness, where the colors are looking, you can't distinguish between red and green. Everything looks basically yellow to you. Okay, so these are both carried on the X chromosome. You need to know them. If in the exam they have to ask you to do a sex-linked disorder and it's about one that you didn't study before, right, like brachydactyly or something, they will give you information about it. They will tell you brachydactyly is an X-linked disorder carried on the X chromosome. Then you must think, aha, now I'm looking at X-linked diseases and disorders. And then when you are doing your alleles, I want you to make sure that the XY looks like that and the XX looks like that. And how do we write it? We put the alleles here. So if you're looking at the hemophilia one, we have this one, and we know that hemophilia is caused by the recessive allele. Sorry, this is not right. Let's just do this again. This is the male. So then if this male is basically affected because it's the recessive allele, and this female, let's make this female unaffected. I just want to show you something. Okay, so the main thing to understand over here is that in a male, we don't have another chromosome to mask the X chromosome. Okay, we don't have another one. The Y cannot mask the X, so it's only one X. Therefore, this male, even if he only has one X chromosome that is recessive, he will have the condition. Whereas the female has to have two recessive alleles to have the condition. In this instance, this female has one dominant allele, which is for normal clotting, and one recessive allele, which is for the disorder. Therefore, the dominant allele is going to mask the recessive allele, and this individual is going to be a female without hemophilia. Okay, please make sure that you write the correct words. Okay, whatever they give you in the question, that's what you need to talk about. If they say it was a hemophiliac male crossed with a non-hemophiliac female, then you need to write those as the phenotypes. Homophiliac male, non-hemophiliac female. Same with the color blindness. Okay, now we go to blood groups. Still monohybrid crosses. Please, I want you to please understand that we are still talking about monohybrid here. Can you see that the alleles have just changed? And now it's a little bit complicated. We're talking about gonosomes, we're talking about X's and Y's. However, it is still monohybrid crosses. We are still looking at one trait. Same with blood groups. Blood groups is about multiple alleles. Multiple alleles does not mean that now we are looking at two genes. We are looking at one blood group. It's basically one gene. Okay, so one gene codes for one blood group. An individual has two alleles for their blood group, and there are three different alleles controlling the blood groups, and then they combine in different combinations to form four blood groups. Okay. Right, so what we need to understand here is this rule. This one is very important. Please make sure you know it. It's the 1, 2, 3, 4 rule, so that you can understand that we have one gene, we have two alleles, we also have dominance and recessive alleles, we also have complete dominance happening here, and we have co-dominance. Okay, I'm going to explain to you why now, just to remind you. So here we've got the blood groups. If you are blood group A, you can have two different kinds of types of genotypes, two different alleles. You can either have homozygous for A, which is this one, or you can have the heterozygous, which is IA small I. Then for blood type B, you can either be homozygous for capital letter B, or you can be heterozygous for I, capital B, and small I. Small I is the recessive allele. You can then be, and can you see these all are complete dominance. Why? Because one allele is completely dominant over the other allele. A is completely dominant over I, and B is completely dominant over I, small letter I. Okay. Whereas in AB, we've got the allele A that is there, and we've got a capital A, and we've got a capital B. So why do we have that? Because this is now co-dominance, because both of them are equally dominant. They are both shown in the phenotype, which means that this person is going to be blood type AB. Then the last one is got to do with complete dominance. Why? Because in the homozygous recessive trait, this trait will be in the offspring, and it's going to be blood type O, and the genotype is homozygous recessive. Right, now we are done with monohybrid crosses. Like I said, please practice. Dihybrid crosses, just remember that now we are looking at two traits that are crossed over here, and they are inherited independently, but there are two traits. So just to sum it up onto this diagram over here. Here we've got a pea plant. The pea plant has many different characteristics. It's got height, it's got seed coat color, it's got flower color, it's got pod color, it's got seed color, okay? It's got seed shape. It's got a whole lot of different traits, but they're all on one individual. So here we are looking at two traits. For example, we're going to look at the seed color plus the pod color. Okay? Those are two traits. Then the next thing I need you to know is Mendel's law of independent assortment. These are when the alleles of a gene for one characteristic segregate independently of the alleles of the gene of another characteristic. Okay. Right, then we've got pedigree diagrams. This is showing you the inheritance. Like I said, I need you to know the different generations. So you need to be able to see here. Please focus over here. The different generation line. This is the first generation, the second generation. How do you look at this? You just look at them and you draw the lines. Okay, the first parents that are in the pedigree diagram, it's a family tree. So the grandparents will be generation 1, their offspring generation 2, then their offspring will be generation 3. Please make sure that you also know that the females are circles and the males are depicted by squares, and that they are shaded and not shaded. These are steps when solving a pedigree diagram. Okay, you can go through them. We have done it in lots of detail. Like I said, we are not going to do paternity testing, but I want to do a question so that we can make sure that we now understand this. So as you can see here, it's a question from the May-June 2023 paper. We actually spoke a lot about this, so I'm just going to go straight to the answer. Basically, they're telling us that it's a hemophilia is caused by a recessive allele on the X chromosome. A woman is heterozygous for hemophilia and marries a man with hemophilia. So it's exactly the example that we did earlier. That's why I'm saying we're going to go through it very quickly now. So please make sure that with the phenotype, can you see there, it says woman without hemophilia. Where do you get it from? You get it from here. But you don't say heterozygous. Can you see that we're not saying heterozygous here? Heterozygous is giving you an indication of what the genotype is. Heterozygous and homozygous are descriptions of genotype. That's a very big misconception that learners have. Okay, so when you are here, you are saying that it's so because you know that it's heterozygous for hemophilia, the female, you're going to know that you must know that it's a woman without hemophilia. So we have the P1 phenotype, which is a woman without hemophilia who marries a man with hemophilia. Okay, and then you're going to have the genotypes. It's going to be X capital letter H, X small letter h, that is going to be crossed with X small letter h and Y. And then we have meiosis forming, we have the gametes. And let's quickly go to what they're asking us to work out. Okay, we'll get there just now. Now we work out the gametes. So can you see that we are separating the gametes? Please make note that we are separating the gametes over here. Okay, this comma shows you that you are segregating them, that they are not together. Please don't write your gametes next to each other in this step. Otherwise, you're going to get it wrong. Okay, then you're going to do your cross or your Punnett square, and then you're going to write down your genotypes and your phenotypes. So let's look. This is one daughter without hemophilia, one son without hemophilia, one daughter with hemophilia, and one son with hemophilia. Okay, but they want to know what is the chance of the couple having a daughter, the percentage chance, what is the percentage chance of the couple having a daughter who is homozygous for normal blood clotting. So if they're going to be homozygous for normal blood clotting, then they're going to have to look like this. Homozygous for normal blood clotting. So they must have two dominant alleles. And do you see anybody here that has two dominant alleles? No. Therefore, there's a 0% chance of a daughter being homozygous for normal blood clotting. Okay? The other thing I want you to know is that this is 6 marks. In this instance, it was actually 7 marks. It was 1 compulsory mark for this because this is what the question is asking you. So this very last point here is very important in any genetic class that you're giving. You're getting. It's giving you the instruction of what you need to work out. Okay? There's no point doing this whole beautiful class and then you don't answer the question. The genetic class is just part of answering the question. Right, we're not going to do this pedigree diagram now, but you do have a worksheet. I've got to mention that we do have a worksheet with all of these same questions in the worksheet. So you can look at the PowerPoint presentation. Please get it from your teachers so that you can actually work through these. You've got the worksheet. You can come through the PowerPoint presentation and get the answers. Your teachers also have the question paper from 2023 that you can also go through. Right, now we go to responding to the environment in humans. Okay, what do you need to know here? You need to know the neurons. You need to know the 3 different kinds of neurons that you have. You also need to know how to draw them. Okay, there are 3 types. You need to know how to draw. And then you've got the brain. You need to know the structure of the brain, the structure of the different parts and the function of those parts. You need to know the spinal cord. You need to know that the brain and the spinal cord are making up the CNS, which is the central nervous system. And then you've got the human eye and the human ear. Okay, very important. These are the main focusing points, but there are also other things. Like I said, we cannot go through all of them. Right, can I just take a 30-second break? Because I just need to 30-second break. Thank you. Thank you. Right. Sorry about that. Let's get back into it. So now we've got responding to the environment. Okay, this is the human nervous system as we spoke about. And this is just summarized for you everything on one page, what I need you to know. You need to know there's a peripheral nervous system, which is made up of your cranial nerves and your spinal nerves. You need to know that we have a central nervous system, which is made up of the brain and the spinal cord, as well as an autonomic nervous system, which is your parasympathetic and your sympathetic. Okay, and then your parasympathetic and sympathetic is now put into this table over here to give you pointers on what happens in the sympathetic nervous system and the parasympathetic. The main thing for you to remember is that your sympathetic nervous system is when you're in a fight or flight response, and your parasympathetic is when you are coming down from that response. Okay, so when you're in a fight or flight response, that's a danger situation, a scary situation, a situation where your heart rate is going to go up, your breathing rate is going to increase, you are going to have dilated pupils, and your wall of your bladder is going to relax, and you're going to have less blood going to your digestive system, because now is not the time for you to digest your food, right? And then your parasympathetic nervous, all you need to know that this is the opposite of the sympathetic. So once your sympathetic has carried out its response, you're going to have to obviously come to a down period to make everything come back to normal, and that's your parasympathetic nervous system. Right, the different parts of the brain, what do you need to know? You need to know the cerebrum, that's involved in voluntary action and memory, the corpus callosum, which connects the left and the right brain, the pituitary gland, which secretes various hormones, the medulla oblongata controls breathing, heartbeat, transmitting nerve impulses, cerebellum balance muscle tension, and hypothalamus control center for hunger, thirst, emotions, and temperature. Okay, so you need to know, these are the main ones that you need to know, and you need to know the functions. They can also ask you application questions on this, so go through some past papers so that you can see what it is that they can ask you. Okay, let's quickly do this activity, because this question, I must say, the nervous systems are not done. I don't know, I don't think learners are struggling with this topic. Whenever this topic is asked in the end of the year exam, they struggle. Let's have a look at the flow diagram. So it shows you the nervous system. It tells you, it's showing you the arrows is divided into two. So you're supposed to know the central nervous system. And what is the other one? I think about it so long. And then the central nervous system is then divided into two parts. And like I told you, it was the brain, and what is the other part? Okay, and then we have the other one, which is the cranial nerves and the other nerves we're going to look at. And then once again, those cranial nerves with those other nerves are going to form another division of the nervous system, and that is the sympathetic and the right. So here we need to identify the component of the nervous system presented by A. So A is the peripheral nervous system. Okay, so how I remember it, central nervous system is in the center. So if I draw a line to the center, it's the brain and the spinal cord. Okay, and then the peripheral nervous system are these. Okay, so it's the ones that are extending from the spinal cord out to the rest of the body. That's your peripheral nervous system. So your peripheral nervous system are sorry, then we've got D. What do they want to know what D is? This is your autonomic nervous system, because here we've got cranial nerves, and then we said that these ones are your so these ones are your spinal nerves, right? Your cranial nerves and your spinal nerves. So this one has to be autonomic. Okay, because you have your cranial nerves and your spinal nerves that are going to form the autonomic system, and then you're going to have sympathetic and you're going to have parasympathetic. Okay, right. So give the letter and name of the component that slows down the heart rate when an emergency situation has passed. Okay, so now the emergency situation has passed. So now they're saying that it's already happened. Okay, so what's the letter in the name of the component? It is E, the parasympathetic nervous system. Because once it's already passed, remember it's already happened. So the sympathetic happened, and now it has to bring you back to normal. You see, this is a very lovely question. If you can do this question, then you understand this. Name the nerve cells that make up the nervous tissue. Those are the neurons. Okay, you should know this from grade 10. You need to know the three: the sensory neuron, the motor neuron, the interneuron. You need to know how to draw them. say 2 ways in which the brain is protected. It's protected by meninges. Okay, meninges are the different layers of the brain. It's protected by the cranium, which is this part, the brain tissue. This is the cranium. It's the part that houses the brain. And it's also protected by cerebral spinal fluid, which is found in the brain. Okay, you need to talk about the brain specifically, so you cannot talk about anything else here, only these 3 things. Then you need to know the reflex arc. You need to know the neurons that are involved in the pathway, and you need to know the difference between a reflex arc and a reflex action. Please know this. Okay, the reflex action is the quick or fast automatic response. That is the response, that is the reflex action, whereas the reflex arc is the pathway that the neurons are going to take to carry out the response. Okay, so those are the difference. Make sure you know the neurons, make sure you know which neuron is going from the stimulus to the receptor. Okay, so we can quickly summarize it. It goes from the stimulus, right? The receptor picks up the stimulus. In this instance, it's a hot object. Okay, then the receptor converts the stimulus into an impulse. That impulse goes via the sensory neuron to the spinal cord. It enters the spinal cord on the dorsal root, and it enters the spinal cord and it makes a synapse with the interneuron in the gray matter of the spinal cord, and then it makes a synapse with the motor neuron. That motor neuron then takes the impulse out via the ventral root of the spinal cord to the effector organ, which is basically your skeletal muscle in this instance, and the muscle is going to contract or withdraw the part that's being stimulated. So it's going to move the hand away so that you don't have further injury, because you've already felt the burn. Now by this point you would have felt the burn because there's another neuron that's going from here to the brain. So you're going to feel that there's pain, but it moves your hand away so that you don't have further injury and you don't burn very badly. Right, then you need to know the 2 sense organs, right? It's the eye and it's the ear. Grade 12, please make sure you know the structure of the eye and the ear. I know that there are many terms, and I know that they are difficult. Okay, they are not easy, but there's different ways for you to go through it. You can do a color-coded diagram maybe, or you can have this put it up on your wall. Life science is about diagrams. If you can just do all of your summary notes now for your June exam, you can put all your diagrams up on your wall, all those study techniques that your teacher told you about that you said, I'm not going to worry about. Now is the time you need to sit and do them. Because if you do them now, you have less to do for the end of the year. If you're going to do a diagram of the eye and you're going to color code them, you're going to color in the sclera one color, the choroid one color. If you're a visual person, that will help you. Then you've done it. Then when it comes to the end of the year, you just take out that diagram and you study it. Okay. So please make sure that for this June exam, you don't just walk into the June exam without studying. Please go through everything. So you need to know the different structures of the eye. So the structure is here in bold, and then we've got the different functions. I'm not going to go through them. You need to know them. Okay? You need to know them, and you also need to know how to compare the structures. For example, you need to compare the sclera and the lens. So you know that the sclera is tough, white, and inelastic. And what do you know about the lens? You know that the lens is elastic, transparent, and it's biconvex. So if you have to compare 2 structures, that's an excellent comparison to say that, okay, the sclera is inelastic, but the lens is elastic. So you need to know the functions of everything, because they're not just going to ask you, give me a function of the lens, or give me the properties of the lens, or the structure of the lens. They're going to ask you to compare the structures of different structures. Right, then the 2 things that learners really get confused with, that I really don't know why, but they do, it's accommodation and pupillary mechanism. Please make sure that you know them. Okay, accommodation, this is when we have near vision or we have distant vision. Please make sure that you know all of these steps off by heart. It comes from Mind the Gap. Please make sure you know them. What I like about this diagram, this GIF over here, is that it shows you how the lens is becoming more biconvex and less biconvex. Okay, so this shape that's showing you here is this is what we call convex. Okay, so it's becoming more convex and it's becoming less convex. So this is more convex, less. So when it becomes flatter, it is less convex. But we don't use the word flatter. We don't use the word round or rounder. Okay, rather use more convex and less convex. So it's in over here where it talks to you about less convex and more convex. That's what learners get wrong. Learners also get these structures incorrect. Okay, They get wrong. They confuse the ciliary muscle with circular muscle. They confuse the suspensory ligaments with other things. Okay, they don't talk about the lens here, they talk about the pupil size. So what I want you to remember is that when we are looking at accommodation, we are looking at the eye accommodating, looking at something that's far or looking at something that's near. So what causes this accommodation? It's about the curvature of the lens. The shape of the lens is going to change. And the shape of the lens is elastic, yes, but something has to cause the lens to move. So it's going to be the suspensory ligaments that are going to be pulled taut. When they are pulled taut, then we're going to be having less convex, and then when they are going to be loosened or they slacken, then we're going to have more convex, because now we are going to have less tension on the lens. Okay, so find ways of, you can even use a mask, okay, to help you with this. You can find different ways, but please make sure that you do know this for accommodation. And they like to ask this question. And then the next question that they like to ask is on pupillary mechanism. Can you see here, this has got to do with the pupil. Okay, so you can see this happening on the external view. When you look at somebody, you can actually see the pupillary mechanism happening. So the pupillary mechanism is a reflex, and it happens in response to bright light and to dim light. Okay, you know this. But what do learners get wrong is that learners don't know that, they don't remember that here we're talking about circular and radial muscles. Okay, and then in the accommodation, we are talking about ciliary muscle and suspensory ligaments. And remember, the another thing that learners get wrong is that suspensory ligaments are ligaments. They cannot contract. Okay, they loosen or they slacken or they become taut. They slacken or they become taut. So I know that they're difficult words to remember, but we need to use those words because those are the correct terms. Okay, we cannot talk about relaxing and contracting when that doesn't happen in ligaments. Okay, you should know that from grade 10. Okay. So we have circular and radial muscles over here, and then they work antagonistically. We know all of this. The main thing I need you to remember here is that here we have a visual picture for our visual learners. Here we have the different words, the sentences, the descriptions that they need to know. And here we've got a mnemonic for learners that are used to that. Okay, so it helps you to say the circular muscle. The pupil constricts and less light enters the eye. Less light enters the eye. Okay, that's what's helping you over there. And then on this one, we have the circular, this is the opposite. So learn one, and then you know that this one is the opposite. Okay, but like I said, it's only practice. You need to know these things, and also you need to know that the circular muscles and the radial muscles are forming part of the iris. When you're talking about this, you must mention the word iris, otherwise you don't get marks, because there are circular muscles in other parts of our bodies. Right, and this is just a difference between accommodation and pupillary mechanism that you can study. Remember that we still have a stimulus here. I just want to mention that quickly. That when we are looking at the nervous system, everything in the nervous system, I want you to remember that we have a stimulus, we have a receptor, okay, and then we are going to have changes that occur. But the part that learners forget is that we have a stimulus. The receptor is going to pick up the stimulus, and the stimulus is then going to be converted into a nerve impulse by the receptor. Then you've got neurons that are going to take the impulse to where they need to go. Learners don't understand this. While the time they understand it in the reflex arc. Then by the time we come to the eye, they forget that we have receptors here. So let's have a look at it. What is the stimulus in for accommodation? It's the change in the distance of the object from the eye. Okay? What is the stimulus in the pupillary mechanism? It's the change in the light intensity. Then what are the parts involved in accommodation? It's the lens, suspensory ligaments, the ciliary muscles. Pupillary mechanism, it's the pupil, which is and the iris has radial and circular muscles. And the main changes are the shape of the lens and the diameter of the pupil. And what brings about the changes is that in accommodation, the suspensory ligaments and the ciliary muscles are what working here. And in pupillary mechanism, we have radial muscles and circular muscles. So these are the ones that learners confuse. Okay, please make sure you know that and you have your ducks in a row. Now, the other thing that you can, this is another question for you that you can do, okay? Now, I didn't do the hearing, eye defects, but please remember that, make a note for yourself that you need to go through your eye defects. I'm also not going to do the ear defects, but you also need to know that, okay? So this is the structure of the ear. You need to know the outer structure, the different parts. I have it here for you with the functions. You need to know the middle ear structure and function. You need to know the inner ear structure and function. and this is the inner ear continued. You need to know all the different parts. Then you also need to know the pathway of sound. Once again, we have a stimulus, we have a receptor. Okay, so when it comes to the pathway outside, this is a very good described question that they can give you, which is like 7 marks. Okay, so you need to talk about the different structures that are involved here. They are the pinna, the auditory canal, the tympanic membrane, the ossicles, the oval window, the cochlear fluid or the cochlea, the organ of Corti, and the auditory nerve. The organ of Corti is the receptor. Okay, the sound waves and the vibrations and the pressure waves, those are the stimulus. These are the stimuli. Okay, then the receptor is going to convert the stimulus into a nerve impulse, and it's going to be taken by the auditory nerve to the brain. Okay, and then what we need to know over here is that you need to know when to use sound waves, when to use vibrations, and when to use pressure waves. The easiest way to remember is that the pinna is going to pick up the sound waves. When it reaches the tympanic membrane, it's going to vibrate. Okay, and that vibration is going to cause the other parts to vibrate. And then when the vibration is going to enter the cochlea is filled with fluid. So we cannot have a vibration. You don't talk about water vibrating. Now we talk about pressure. Okay, so now we talk about pressure waves. Then those pressure waves are going to stimulate the organ of Corti to convert the receptor, convert the stimulus into a nerve impulse, and then it's taken by the auditory nerve to the cerebrum. We don't say brain, we say that it goes to the cerebrum. Please remember that. This is another mnemonic for you to remember so that you can remember the different parts that you need to know. Then there are 2 ways that the inner ear regulates balance. Okay. The main thing that I want you to understand for this one, grade 12, is that this part remains the same when it comes to balance. It's these 2 parts here, right? Number 1 and number 2, which differ. Because the receptors differ, because the stimulus differs. That's what learners don't understand. This is the stimulus. This is the stimulus here. That's the first line. That's the stimulus, and this one here is the stimulus. Okay? So this is when we have a change in in number 1, we have a change in direction and speed. Okay? So that means that I am running. Okay, I'm running or I am moving forward. Therefore, my head, the direction is moving. Okay, the direction is changing, and its speed. So if I'm running, and then all of a sudden I turn, I'm changing direction, right? Okay, so this is when our receptor is going to be stimulated, which is the crista. Where are they found? You need to know that the ampulla, it's found in the ampulla, in the semicircular canals. But in this one, we have change in the position of the head. Let me show you an example. Like my head is here. If I'm going to put my head down or put my head up, it's in response to gravity. Can you see? Right? So if my head is going to change in that direction, then I'm going to have my macula, which are stimulated. And that's in the sacculus and the utriculus. Then, whether it is the crista or whether it's the macula, what's going to happen? Both of those receptors are going to convert the stimulus into an impulse, and it's going to be sent by the auditory nerve to the cerebellum. It then sends an impulse to the skeletal muscles to restore balance. Okay, that's what we basically talk. We talk about the skeletal muscles in particular that's going to be affected here, and it's to restore the balance. So it's when you are running or something and all of a sudden you trip. What prevents you from falling is now this cerebellum. Okay. Now, this is just a summary to remind you what the organ and the receptor is, but I did this in a lot of detail, so I'm not going to do it with you now. Okay. Then we are quickly going to do one activity because I think it's important for us to do the structures of this for those of you that did not get it. Okay. The diagram below shows part of the middle and the inner ear. Name the receptors that are stimulated by a change in the position. Okay, first we're going to do this one. Identify part F. So here we've got diagram 1 and diagram 2. So what you first need to do, you need to ask yourself what's happening in diagram 1, what's happening in diagram 2. Diagram 1 is showing us the middle ear, and diagram 2 is showing us the inner ear. Okay, now they want to know what's F. So you know it is the semicircular canals. How do you know? We've got those loop structures, and they're at 90 degrees to each other. Give the collective term for the bones B. So those bones are the ossicles. Remember, you should know them. Also, separately, you should need to know that they are the malleus, the incus, and the stapes. And together, they are known as the ossicles. Okay? They are the smallest bones in the human body. Give the letter and name of the structure that equalizes pressure between the outer and middle ear. Now, what learners do is they see this and they get confused. They just write the name because they know the word. but they're asking you for the letter and the name. It's very important that we read the question. So we go to the letter and what's the name. They're talking about equalizing pressure between the outer and the middle ear. So what is it going to be? It's going to have to be the D, which is the eustachian tube. So you're going to say D, eustachian tube. If you only say eustachian tube, you only get 1 mark. If you just say D, you're going to get 1 mark. Okay, so please read the question. The second one is they want to know the letter and the name of the part that creates pressure waves in the inner ear. So this is here your pressure waves, and it is caused by the oval window. So this is C over here. This is the oval window. Now don't get confused. This one's the oval window. The bottom one here is the round window. It's the oval window because the oval window is going to take this vibration of the ossicle. It's going to go through the oval window, and then you're going to have a pressure wave that is now going to be formed in the inner ear. Okay, and then we have the name and name the receptors that are stimulated by a change in the position of the head. Okay, so now remember what I said, the position of the head is the maculae. Don't be alarmed by the spelling here. Maculae is the plural, and then the direction and speed of movement in the head is got to do with the crista. Okay, that's why I said we have to do this activity to show you that here we're talking about the structures, we are talking about the different, we are talking about balance, and this is actually found in section A in your question paper. Okay, so like I said, that's all for us for today, but I just want to go through this. Let's sum up quickly because we did a lot. Okay. So the main take-home message that I want to tell you is that we try to cram in a lot of important things that you need to know. Okay, but the important thing is that you practice. You study in advance and that you practice. Okay, you practice past paper questions because once you've studied, they're going to ask you. They're not just going to ask you for the function of something. You saw the question. You're going to need to apply your knowledge. So make sure that you know your content well. What are the things to bring in an exam room? You shouldn't be caught without these: a compass, a ruler with numbers, a protractor, a calculator, a pencil, and extra pens. Okay, why do you need these things? Why do you need the compass and the protractor? Because remember, you are going to be asked to draw a graph. Okay, so you can either be asked to draw a pie chart, or you can be asked to draw a bar graph, or a line graph, or a histogram. And if you have a pie chart to draw, you need a compass and you need a protractor. Otherwise you can't draw the circle and you can't work out the sectors, okay, the degrees. You need a ruler with numbers. Why? Because you're going to be asked to draw things. You're going to be asked to read off from graphs. Okay, you're going to have to use a ruler, as Samantha said, to do your DNA profiling. So a ruler is very important. And then your calculator for calculation. We didn't get to do an investigation today, but please remember that you are going to get an investigation question. It can be on any topic. I cannot tell you which one it's going to be, but there is going to be an investigation question. You probably are going to be drawing a graph. If you're not going to draw a graph, you are going to read off a graph. Okay, you will be seeing a monohybrid cross in some form or other. Right. So please make sure that you go through everything, go through this lesson. Please remember that we also have the worksheet, and good luck. That's all from us. Thank you.